Good afternoon to all the NEET aspirants. This is your Prasanna Ma'am from NEET World, and yes, today we are going to discuss the NEET previous year questions from the chapter Molecular Basis of Inheritance. This is the second session. So far we have dealt with NEET 2025 and NEET 2024 questions of this chapter. Today we are going to look into NEET 2024 to NEET 2020 questions from this chapter, including concept clearance beta. So without much delay, let's get started. And the first question, which is from NEET 2024, and this question is of match the following type. Okay. So here if you look into first RNA polymerase 3. We have 3 RNA polymerases in a eukaryotic nucleus, RNA polymerase 1, 2 and 3. RNA polymerase 3 is involved in snRNA and tRNA, along with that 5S RNA synthesis. Fine. So RNA polymerase 3 is involved in the synthesis of snRNA and tRNA. That is option 4. And then termination of transcription. Transcription termination is by Rho factor. Okay, termination process in the transcription is by Rho factor and initiation is by sigma factor. Splicing of exons is by SNRPs. These are small nuclear ribonucleoproteins. Okay, small nuclear ribonucleoproteins which has RNA and proteins in it. Okay, so splicing of splicing of introns include what? SNRPs. This is the process mediated by SNRPs in post-transcriptional event in a eukaryotic cell. And Tata box during transcription, this is present in the upstream of the coding sequence in the promoter region, close to the promoter region, which is option 2. So here if you have to look into the right answer, RNA polymerase 3, which is option 4, involved in the synthesis of snRNAs and tRNA. A is 4 and B if you 3, termination of transcription, that is by Rho factor. Splicing of exons is by SNRPs, which has RNA and proteins. C is 1. Tata box, which is present at the upstream in the promoter region. So D is option 2. So option 1 will be the right answer. Next question you look into statement based question. Given below are the 2 statements regarding RNA polymerase. So again the question is on RNA polymerase in NEET 2024. So here if you look into statement 1, in prokaryotes RNA polymerase is capable of catalyzing the process of elongation during transcription. Yes, RNA polymerase is capable of catalyzing the process of elongation during transcription, true. And here in statement 2, RNA polymerase associate transiently with Rho factor to initiate transcription. To initiate transcription, for initiation, RNA polymerase will bind with sigma factor, which forms out to be a holoenzyme. Fine. And for termination, at the time of termination, for termination during the process of transcription, RNA polymerase will bind to Rho factor. Fine. So here if you see, statement 1 is true and 2 is false. Statement 1 is true and 2 is false. Option 1 is the right answer. This question you see, which of the following is a nucleotide? Nucleotide is the component which has sugar, nitrogen base and phosphate group or phosphoric acid. It should have sugar, nitrogen base and phosphate group. So the example over here amongst these 4 options, the nucleotide will be adenylic acid, which has ribose sugar and the adenine group with phosphoric acid or phosphate group, that is adenylic acid. Uridine and guanosine, these 2 are nucleosides. Uridine and guanosine, these are what? Nucleosides. Fine. Guanine is what? Nitrogen base. Guanine is nitrogen base. Clear, right? So here what is the nucleotide ma? Option 2 is the right answer, that is adenylic acid. Next question you see, again a statement based question appeared in the year NEET 2024. Here if you see, given below two statements, statement 1, in eukaryotes there are 3 RNA polymerases in nucleus in addition to RNA polymerase found in the organelles. True. In a eukaryotic cell, inside the nucleus we have 3 RNA polymerases. What are they ma? RNA polymerase 2, RNA polymerase 3, these are present inside the nucleoplasm. Inside the nucleolus, RNA polymerase 1 is present. RNA polymerase 1 is involved in the synthesis of rRNA, RNA polymerase 2 is involved in the synthesis of mRNA or heteronuclear RNA, RNA polymerase 3 is involved in the synthesis of rRNA or snRNA. Clear, right? So these 3 are present in the nucleus of eukaryotic cells. In the organelles like chloroplast and mitochondria, which are semi-autonomous or self-duplicating organelles, right ma? These organelles will have only RNA polymerase, like how you see in a prokaryotic cell. So apart from RNA polymerase which is present in the organelles, in eukaryotes there are 3 RNA polymerase inside the nucleus. So statement 1 is true. In statement 2 if you look into, all the 3 RNA polymerases in eukaryotic nucleus, they have different roles. Yes, each of them is synthesizing different molecule. So statement 2 is also true. So both the statements are right over here, both statement 1 and statement 2 are correct. Option 3 is the right answer. Next question. Match the following type of question again. Parallelly even you can answer ma, and if any doubt anywhere you can drop a message in the chat box, I will be answering it. So here in the match the following type of question, if you see, first histones. Histones are positively charged basic proteins, right? These are the main function of histone proteins is to help in DNA packaging or DNA wrapping. Second is nucleosome. Nucleosome is well accepted model for DNA packaging in eukaryotic cells. In nucleosome what you see, DNA is wrapped around the histone octamer and around 200 base pairs are present in the nucleosome DNA. Euchromatin. Euchromatin is loosely packed chromatin, which is option 1. C is option 1. Heterochromatin is densely packed chromatin which is inactive. Densely packed chromatin which is inactive. Euchromatin is loosely packed which is active. Fine. So here if you see histones, positively charged basic proteins, this has what proteins ma? Arginine and lysine. And then nucleosome, where DNA is wrapped around the histone octamer with 200 base pairs of DNA. Euchromatin is loosely packed chromatin, heterochromatin is densely packed chromatin, heterochromatin is inactive, euchromatin is active. Look into the options carefully ma. A is 3, B is 4, C is 1 and D is 2. Option 4 will be the right answer. Okay. Next question. 10 E. coli cells, this is application based question, look into that keenly. 10 E. coli cells with N15 isotope of double stranded DNA are incubated in the medium containing N14 isotope of the nucleotide. After 60 minutes, how many E. coli cells will have DNA totally free from N15? Firstly, this experiment is from, this question is from which experiment ma? Michelson-Stahl's experiment, which is an experimental proof for semi-conservative method of DNA replication. Okay, semi-conservative method of DNA replication. This question is a solid proof for that. Okay, and here we need to apply the semi-conservative model of DNA replication, wherein after each replication the daughter cells will be having one parental strand and one daughter strand of DNA. So here if you see, The parental strand here 10 E. coli cells he is saying. So, let us take for 1 E. coli cell first. It has both the strands of N15, N15. Just a second. Yeah, both the strands are of N15 and N15. Fine. They are incubated in the medium containing N14 nitrogen solution. After 60 minutes he is saying in the question. One generation time in the E. coli is 20 minutes. One generation time is 20 minutes. So, 60 minutes means it is around 3 generations. 60 minutes means 3 generations, right. So, first 20 minutes you will get 2 hybrid strands, one parental strand and one daughter strands, 2 hybrid strands you will get of N15, N14, N15, N14. When these hybrid strands are further incubated, when they are further incubated in N14 solution for 20 minutes, okay, for 20 minutes, you will get again 2 hybrid strands and 2 light chains. Fine. When they are further incubated, for 20 minutes, same the case, you will get 2 hybrid strands, 6 light chains, 6 light chains. Just count on to the parental strands, then you will get how many daughter strands are possible. For each strand there will be one daughter strand, right, after the generation time. So, here there are 2 heavy isotopes. So, after 20 minutes you will get 2 hybrids and 6 light chains are there. So, after 20 minutes you will get 6 light chains. So, if you take 1 E. coli cell and incubate it for 60 minutes in N14 solution, at the end you are getting 6 light chains. Same the case if you repeat with 10 E. coli cells, 10 E. coli cells for 60 minutes. After 60 minutes you can see 60 cells which are totally free from N15 isotope. Clear, right? Next question. In an E. coli strain, I gene gets mutated and its product cannot bind the inducer molecule. Okay. This is also application based question from Lacoperon model. Very important. Listen carefully. In an E. coli strain, I gene gets mutated and its product cannot bind with the inducer. I gene is mutated. What is its product? Repressor is the product. The repressor cannot bind to the inducer. If growth medium is provided with lactose, what will be the outcome? Anyhow, the repressor is unable to bind with the inducer, that is lactose. So, even if you supplement the growth medium with lactose or not, the repressor is free and repressor will easily bind with the operator. When repressor is binding with the operator, then RNA polymerase cannot bind with promoter region. Okay, understood? I hope you understood the question. Once again I will repeat in the question he is saying like in an E. coli strain, I gene which is repressor, I gene gets mutated and its product cannot bind to the inducer. I gene gets mutated, its product is nothing but repressor is the product of I gene. So, it cannot bind with the inducer, that is lactose. When it cannot bind with lactose, it binds with the operator. In continuation in the question he is mentioning like if growth medium is provided with lactose, what will be the outcome? If growth medium is provided with lactose also, repressor cannot bind with lactose. In that case it binds with operator and RNA polymerase cannot bind with the promoter region and Lacoperon model lactose metabolism is inhibited. In that case Z, Y and A genes will not be translated. Okay, though you supplement the medium with lactose or not, if I gene gets mutated and repressor is not binding with the inducer, Z, Y and A genes will not be translated. Option 2 will be the right answer over here. Next question you see read the following statements and choose the set of correct statements. Correct statements we have to choose. This is a multi-statement question. Okay. First statement, Euchromatin is loosely packed. This is third time we are looking the question on the euchromatin and heterochromatin. That much important it is. Euchromatin is loosely packed chromatin, true. Heterochromatin is transcriptionally active. Heterochromatin is transcriptionally inactive, okay, not active. Histone octamer is wrapped by negatively charged DNA in the nucleosome, true. Histones are rich in lysine and arginine, correct. That is the reason it is positively charged, and because it has basic proteins. A typical nucleosome contains 400 base pairs of DNA. No, a typical nucleosome contains 200 base pairs of DNA. Fine. So, here A is true statement, C is correct and D is correct. Fine. So, here what is the correct set of statements, ma? A, C and D. A, C and D. Option 1 will be the right answer. This is the question which has appeared in NEET 2022. Next question you see, NEET 2023, 2 statement based questions only. First you look at this. Statement 1, in prokaryotes, the positively charged DNA is held with some negatively charged proteins in the region called as nucleoid. First of all, DNA is not positively charged, DNA is negatively charged, and they are surrounded by with some positively charged non-histone proteins, not histone proteins in a prokaryotic cell. So, statement 1 is wrong. Statement 2, if you see, in eukaryotes, the negatively charged DNA is wrapped around positively charged histone octamer to form a nucleosome, true. Statement 2 is correct. Statement 1 is incorrect and statement 2 is true. Option 3 will be the right answer. Next question you see, again statement based. Statement 1, RNA mutates at a faster rate. Yes, RNA mutates at a faster rate because this is unstable. Okay, RNA mutates at a faster rate because it is unstable. Fine. Statement 2, virus having RNA genome and shorter lifespan mutates and evolve faster. Yes, it has shorter lifespan and it evolves faster. So, both the statements are true over here. What is the right answer, ma? Statement 1 and statement 2, both are correct. Option 4 is the right answer. Next question. Yeah. Unequivocal proof that DNA is the genetic material was proposed by whose experiment? Frederick Griffith, he gave us transformation principle. Wilkins and Franklin gave us X-ray diffraction. Avery, MacLeod and M.C. Carte has proved that DNA is the genetic material. Hershey and Chase conducted experiments on bacteriophages and said that DNA is the genetic material. But unequivocal proof is given by whom? Avery, MacLeod and M.C. Carte. Next question you see, match the following type again. Gene A. First you look into Lacoperon model because the question is asked from Lacoperon model wherein you have promoter, operator and 3 structural genes ZYA. Okay. So, here A codes for what, ma? Trans-acetylase. Trans-acetylase. Y for permease. Z codes for beta galactosidase. Fine. So, here A is coding for trans-acetylase, that is option 2, Y permease and I is repressor protein, Z is coding for beta galactosidase. So, here A is 2, B will be 3, C is 4 and D is 1. Option 1 is the right answer. Next question you see, ma? Statement based question only. What is the role of RNA polymerase 3 in the process of transcription in eukaryotes? We have already discussed the functions of RNA polymerase 1, 2 and 3. 3 function is mainly it transcribes what? tRNA, 5S RNA and snRNA. mRNA, it has no role in mRNA because mRNA synthesis is done by what? RNA polymerase 2. So, here option 1 will be the right answer. And look at here NEET 2021 question. If adenine has adenine has 30% of the DNA molecule, what will be the percentage of thymine, guanine and cytosine? Such types of questions are easily decoded by Chargaff's rule that number of purines are equal to number of pyrimidines, and adenine pairs with thymine by 2 hydrogen bonds, guanine pairs with cytosine by 3 hydrogen bonds. As such, number of purines will be equal to number of pyrimidines. So, if there are 30% of, if there is 30% of adenine, thymine also will be how much ma? 30%. So, totally it is 60% out of 100%. Out of 100, 60% is adenine and thymine. Rest is what? 40%. Out of 40%, 20% will be guanine and 20% will be what? Cytosine, where thymine is 30%, guanine is 20 and cytosine is 20. Yes, correct. Good. Samadhita, true ma, correct. Next, this question you see Vita, which of the following RNAs is not required for the synthesis of proteins? Okay. For the synthesis of protein, we need tRNA, mRNA and rRNA. rRNA is the structural ribosome, mRNA carries the genetic codon, tRNA will brings out the amino acids. So, 1, 3 and 4 are required. Silencing RNA is not required. Option 3 is not required, that is siRNA, silencing RNA is not required, okay, for the synthesis of protein, option 3. And then here you see which of the following statements about histones is wrong, okay, about histone, which of the following statement is wrong. The pH of histone is slightly acidic, wrong, the pH of histone is slightly basic, not acidic, okay. Histones are rich in amino acids like lysine and arginine. Histones carry out positively charged in their side chains, true, and histones are organized into a unit of 8 molecules. Yes, they are a unit of 8 molecules of 4 pairs. What are they ma? H2A, H2B, H3, H4. Okay, these are 4 histone proteins of 2 sets. So, total it is 8 molecules. So, here which is the wrong statement? Option 1. Very good ma. Look at this question. Try to answer this beta. The codon AUG codes for methionine and phenylalanine. Wrong. AUG codes for methionine and this also acts as an initiator codon. But this does not code for phenylalanine. This is coded by UUU. Okay. Statement 2. AAA and AAG, both codons code for amino acid lysine, okay, both the codons are coding for amino acid lysine, fine. So, here if you look into the right answer, statement 1 is wrong and 2 is true, option 3 will be the right answer. This question you see, NEET 2021, try to answer this. Complete the flow chart on the central dogma of molecular biology. First is A. This is what ma? DNA replication. Okay. First is replication. Replication is followed by transcription. Replication followed by transcription. Then mRNA to proteins, right? This is what C is translation. And lastly, ultimately that is the synthesis of proteins. Fine. So, here if you see, A is replication, B will be transcription, C is translation and D is proteins. Replication, transcription, translation and D is proteins. Option 2 will be the right answer. Last questions of today's session. Look into that carefully ma. The first phase of translation is what? First tRNA should get charged. That is called as what? Amino acylation of tRNA. This is the first step of translation. Okay. And then binding of mRNA to ribosome and third will be recognition of anticodon. The very first step is what? Amino acylation of tRNA. Next question of NEET 2020. Name the enzyme that facilitates opening of DNA helix during transcription. During transcription, if DNA helix opening during replication, then our answer will be DNA helicase. During transcription, so our answer should be what? RNA polymerase. RNA polymerase along with the sigma factor will help in opening of DNA double helix during the transcription process. Last question you see ma, which of the following statements is correct? Which of the following statements is correct? Adenine pairs with thymine through one hydrogen bond. Wrong, we all know that adenine pairs with thymine with 2 hydrogen bonds. Adenine pairs with thymine with 3 hydrogen bonds. Wrong. Adenine does not pair with thymine. No. Adenine pairs with thymine with 2 hydrogen bonds. Option 4 will be the right answer. Okay. So, in our next session, we will be planning to exercise the advanced set of questionnaire targeting NEET 2026 from the chapter molecular basis of inheritance. So, it is a very useful session ma to all the aspirants of NEET 2026. Don't miss that at all. And that's all for today's session. Thank you.